Today we'll be discussing the different steps in mitosis. We will start our discussion at prophase. Now it is important to note here that the DNA has already been replicated. As you can see here, the centromere and the arms. Uh, each chromosome already has two chromatids. Now you can still see that the nucleolus is present here. You can see that the centrosomes are present here, and you can see some formation of asters and other microtubule. Another thing to notice here is that the cell membrane is represented in blue. As we get closer to approaching metaphase, I will notice that the nucleolus has dissipated. That would normally be there. Eventually, before we get to metaphase, or to the later metaphase, we're going to see that these actually start to connect. You're going to see connections of the kinetochores from both sides, and they're specifically going to attach onto the centromeres. As we get into the later metaphase, we're going to see what happens.
During metaphase, all chromosomes align in the middle of the cell at the metaphase plate, which is represented by the red line, and the centrosomes are located at opposite sides of the cell at the spindle poles, which are attached to the poles with asters, which are these smaller green dots. The darker green, non-kinetochore microtubules go between to attach to each other, which we can see by these, and the lighter green attach to the centrosomes, which are the orange dots in between.
So we're now in anaphase, and we often refer to this phase as the away phase, because as you can see by our picture, the chromosomes are being pulled apart from each other or away from each other. So the kinetochore microtubules that are in light green pull towards opposite sides as they shorten, and then the non-kinetochore microtubules that are in dark green grow to elongate the cell and prepare for the next step. Lastly, it's important to note that the number of the chromosomes doubles in this phase.
Telophase is the last phase of mitosis. In this phase, the mitotic spindles are on opposite ends, right here. And the cleavage furrow starts to form in between the cells, so it would be in between right here. We can see that the chromosomes and spindle fibers start to decondense while the nuclear membrane starts to reform. After this phase, cytokinesis follows, which is just the division of the cytoplasm, which is the separation of the two cells.